Okay, so the last topic we're going to talk about in this unit is cellular respiration. And cellular respiration occurs in autotrophic and eukaryotic heterotrophic organisms as well. And this gives you an overview of the process. It takes place in three main steps: glycolysis, then pyruvate oxidation, citric acid cycle, and then the oxidative phosphorylation of the electron transport and chemiosmosis. So we want to talk a little bit about catabolic and anabolic pathways. Catabolic pathways occur when molecules are broken down and their energy is released. You'll need to know about fermentation and aerobic respiration. Fermentation is the partial degradation of sugars that occurs without the use of oxygen, and aerobic respiration is the most prevalent and efficient catabolic pathway in which oxygen is consumed as a reactant along with the organic fuel. So these catabolic pathways yield energy by oxidizing organic fuels. So carbs, fats, and proteins can all be broken down to release energy in cellular respiration. Glucose is the primary molecule used in cellular respiration, and it's the one we'll refer to the most often. The standard equation for this cellular respiration is 1 glucose molecule plus 6 oxygen yields 6 CO2, 6 water, and an abundance of energy in the form of ATP. Energy is released, or exergonic, from glucose to phosphorylate ADP to ATP in cellular respiration. Cellular respiration's reactions are a type of oxidation, reduction, or redox reactions. Redox reactions are where electrons are transferred from one reactant to another, called oil-rig. Okay, that's the mnemonic that we use to remember it. So oxidation is the loss of one or more electrons from reactant. So we call it oil because in oxidation, electrons are lost. In reduction, it's the gain of one or more electrons. So we call it rig because reduction is gaining an electron. In cellular respiration, electrons are stripped from glucose in this redox reaction. So during cellular respiration, a glucose molecule is gradually broken down into carbon dioxide and water. Along the way, some ATP is produced directly in the reactions that transform glucose. Much more ATP, however, is produced later in a process called oxidative phosphorylation. And this is powered by the movement of electrons through the electron transport chain, a series of proteins embedded in the inner membrane of the mitochondria. If you remember, we had one of those in photosynthesis as well. There are three main stages of cellular respiration. You have glycolysis, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation. And we're going to get into these three steps. So glycolysis is where we harvest chemical energy by oxidizing glucose to pyruvate. Remember, we're oxidizing, so that's oil, so we're losing electrons. So we take the 6-carbon glucose and we break it down into 3 carbon pyruvate molecules through a series of steps. This process does not require oxygen. What you do need to remember from this process is going from a 6-carbon glucose to 2 3-carbon pyruvates. It yields 2 pyruvate molecules, 2 ATP molecules, and 2 NADH. There is an energy investment phase and an energy payoff phase, and that's what gets us our net products. And there's an excellent video on the collection of cellular respiration videos in your table that can explain to you exactly the details of this if you want to go into it. But what you need to mainly know is that glycolysis yields 2 pyruvate, 2 ATP, and 2 NADH. The process of how it occurs, you don't have to really know. So like I was saying, there's an ATP consuming and an ATP producing. In the ATP consuming phase, 2 ATPs are used up, and this helps destabilize glucose and make it more reactive. So the glucose comes in, we use 2 ATPs to destabilize the molecule. Then in the ATP producing phase, 4 ATP are produced right here for a net gain of 2 ATP since we used 2 ATP above. And 2 NADHs are produced. And those are going to be used in the electron transport chain to produce more ATPs. So again, if you look down here, this is our net gain from glycolysis. 2 pyruvates, 2 ATPs, and 2 NADHs, plus 2 waters. Most of the potential energy of the glucose molecule still resides in the 2 remaining pyruvates, and these are what are going to move into the citric acid cycle. So after pyruvate is oxidized, the citric acid cycle completes an energy-yielding oxidation of organic molecules. So first we take those pyruvates and we oxidize them into acetyl coenzyme A. The transport protein moves the pyruvate from the cytosol to the matrix of the mitochondria where the rest of cellular respiration is going to take place because remember the job of mitochondria is to make ATP. So in the matrix of the mitochondria, an enzyme complex catalyzes 3 reactions. A CO2 molecule is removed and electrons are stripped from the pyruvate, and a coenzyme A joins with the 2 carbon fragment to form acetyl coenzyme A. So 2 acetyl coenzyme molecules are produced per glucose molecule. So each acetyl coenzyme A now enters the citric acid cycle. So you can see in my diagram we have the pyruvate. Remember, 2 pyruvates come in for every glucose, and a carboxyl group is removed from the pyruvate, releasing the carbon dioxide right here. Then an oxidation reaction occurs, and NAD is reduced to NADH, and that's where we add on the coenzyme A product, and we end up with the acetyl group is transferred to the coenzyme A, resulting in acetyl coenzyme, and that's going to go into the citric acid cycle. Now, in the citric acid cycle, glucose is broken down and is completed with CO2 released as waste. Each turn of the cycle requires input of 1 acetyl coenzyme A. So the cycle has to make 2 turns in order to, before glucose is completely oxidized. So what's produced in each turn? 2 CO2, 3 NADHs, 1 FADH, and 1 ATP. 2 pyruvates are yield a total of 4 carbon dioxides, 6 NADHs, 2 FADHs, and 2 ATPs. Because remember, it has to make 2 turns of the cycle. So 1 acetyl coenzyme goes in, and then a second one goes in. And those acetyl coenzymes originally came from the 2 pyruvates, which originally came from the 1 glucose molecule. At the end of the citric acid cycle, 6 original carbons in glucose have been released as CO2. Only 2 ATPs have been produced. So where's all the energy then? Okay, this is where it comes from. NADH and FADH are electron carriers produced in the citric acid cycle. And these electron carriers will be used in our electron transport chain to produce the rest of the ATPs. So what you need to remember the citric acid cycle is each pyruvate yields 4 NADHs, 1 NADH, 1 FADH, and 1 ATP. And we're going to do that 2 times. Then we go into the oxidative phosphorylation or chemiosmosis to couple the electron transport to ATP synthesis. So there are thousands of electron transport chains embedded in the inner membrane of the mitochondria, and each is composed of 3 transmembrane proteins that work as hydrogen pumps and 2 carrier molecules that transport electrons between hydrogen pumps. So you can see in the image, we've got our hydrogen pump here, our hydrogen pump here, and a hydrogen pump here. And then they are going to, and then we have our carrier molecules right here and right here. And their job is to move the electrons through the electron transport chain to get it to the ATP synthase, where we're going to convert ADP into ATP. The electron transport chain is powered by electrons from the electron carrier molecules NADH and FADH2. As the electrons flow through the electron chain, the loss of energy by the electrons is used to power the pumping of protons across the inner membrane. At the end of the electron transport chain, the electrons combine with two hydrogen ions and work to form water. Oxygen is the final acceptor, and it's necessary for the ATC to work. The hydrogen ions flow back down their gradient through the ATP synthase, just like they did in photosynthesis. And then the proton motive force is in place because the inner membrane of the mitochondria is impermeable to hydrogen ions. So like water behind a dam, with its only exit being a spillway, the electrons are held behind the inner membrane with their only exit through the ATP synthase. This process is referred to as chemiosmosis. Chemiosmosis is an energy coupling mechanism that uses energy stored in the form of a hydrogen gradient across a membrane to drive cellular work. The electron transport chain and chemiosmosis compose oxidative phosphorylation. This specific term is used because ADP is phosphorylated into ATP and oxygen is necessary to keep the electrons flowing. The ATP yield per molecule of glucose is 30 to 32 ATP. Oxidative phosphorylation produces 26 to 28 of the total molecules. You may notice that this number is less than you might have learned elsewhere, but it more accurately reflects the current biochemical analysis. So you need to be able to indicate the mitochondrial features that are important to establish the proton gradient. So this diagram, I would practice drawing it out and knowing the steps that everything goes through. The electron transport chain is a series of proteins and organic molecules found in the inner membrane of the mitochondria. Electrons are passed from one member of the transport chain to another in a series of redox reactions. Energy is released in these reactions, is captured as a proton gradient, which is then used to make ATP in a process called chemiosmosis. Together, the electron transport chain and chemiosmosis make up the oxidative phosphorylation. So, that's all if oxygen is present. If oxygen is not present, then we have fermentation and anaerobic respiration. Anaerobic respiration is the generating of ATP without oxygen, but it uses an electron transport chain. This can happen in certain prokaryotes. Fermentation is an expansion of glycolysis in which ATP is generated by substrate level phosphorylation. Fermentation consists of two things: glycolysis, which nets two ATP molecules on its own. Oxygen is not needed to accept the electrons, and NAD is the acceptor, and reactions that regenerate NAD. There are two types of fermentation. You have alcohol fermentation and lactic acid fermentation. Alcohol fermentation is where you go from pyruvate to ethanol, releasing carbon dioxide and oxidizing NADH in the process to create more NAD. Lactic acid fermentation is where pyruvate is reduced by NADH, and lactate is formed as a waste product. And we have the two diagrams that kind of go through the process of how this works. Fermentation and anaerobic respiration enable cells to produce ATP without the use of oxygen. There are special organisms, one called facultative anaerobes, that can make ATP with aerobic respiration if the oxygen is present, but they can switch to fermentation if the oxygen is not present. Muscle cells can do this. And then you have obligate anaerobes, and they can't survive in the presence of oxygen, so they have to do fermentation. So what do you have to know? Okay, you don't have to memorize each individual step and what is created and what is used. You need to understand the process as a whole and focus on the energy transfers, what goes in and what comes out, not the exact reaction that create the products. You don't need to know the names of the enzymes involved in the process. So you generally need to know what's in this diagram, okay? You need to know a